Okay, thanks, Ben. So as Ben said, I'm Priya Ranjan at Oak Ridge National Lab. And this work together we did with Sanj and Bluth, this presentation. And so as Ben said, this is part of a series of presentations that we are doing in KBase related to KBase samples. The first one was the overview, where we talked about the big picture of the samples, what samples are, what are the future ambitions of the sample, what you can do with it right now, what you can do within the future. So I won't go into too much detail into sort of the overview. And Ben already pointed to a YouTube video where you can go and look at the sort of the last presentation. But I'll give you a small overview. And so just the topics that I'll cover today is the overview of samples, sample management in KBase, the apps that exist for importing samples. How can you bring samples from external repositories? What are the repositories we support? How do you prepare sample metadata file from scratch if you don't have something existing in external repositories? The location of sample documentation, Ben already showed that, the templates that we are supporting and control vocabulary. And this is supposed to be a little bit more details than the overview. So I have some videos that show you how to go through the process and then understand errors as you go while you are importing and uploading samples into KBase. Now the SampleSet landing page is a way to explore the samples and find linked data. So I'll show that. Then let's say if you have made a mistake, how do you update your samples and how are things versioned? I'll give you a sense of that. And I'll also show you how to have control over your samples, who can edit samples, who can link data to samples. So we have mechanism to have that sort of control. And then future webinar topics and the team. We have a nice team that helped build the system in KBase. So this is from the last the webinar that we had, and it's basically what is a sample? It's basically a physical material that you sort of collect at any point of time, and then you attach attributes to it, like biogeochemical measurements of the environment. The material it belongs to, and that comes from environmental ontology, or the biome that it belongs to, which helps make the data fair, which is that only you, rather than the person creating the data, others can also understand it. And it has all of that information in it. The formats that we currently support, I must say that, and it was said in last webinar as well, right now we are at version 1.0 of the samples. So this may not have all the features that you're looking for, but it's pretty exciting. It has a lot of features that we have intended to build for a long time. So currently, though, the formats that we support are from Earth sample registry system at SESAR, Enigma, and KBase. And then the link for documentation is here. The idea is there that you have these sample attributes, and then you can connect measurements on those samples within KBase. And again, so in KBase, we are expecting the samples, especially the environmental samples, to have geographic location, temperature, collection information, environmental information, material depth. And these can then be, the samples can then be linked to the measurements on those samples, such as chemical abundance in those samples, or if the metagenomic sequence has been done, then that's linked to that. Or if you have amplicon abundance, it gets linked to that. But before you can do any of these analysis, full analysis in KBase on these data sets and get insight into your data, the first place obviously is to upload samples, and then you can link things. This is sort of a new thing in KBase. Before this, we didn't have this concept of samples. And so it's a completely new thing, but it's a way to organize all of your data. And soon, I think every piece of data that comes to the system will have some linkage to a sample. So again, it helps you do a lot of analysis, and we have some pretty ambitious goals there. But right now, what can you do once you upload samples in KBase? You can upload amplicon tables and connect to samples. And that's the only way to upload it. You need samples to be able to do that. But then you can visualize community composition based on sample data, see that. You can make functional predictions for individual taxa and communities. You can examine relationships between microbial community composition, environmental factors. Like I am interested in plant microbe interaction. And so I was able to upload my data sets in KBase, and I was able to figure out if there is a relationship between host genotypes and microbiome profile, or if there is a relationship between chemical abundance and microbiome profile, or the soil composition, is that the major deciding factor in microbiome profile? So there are apps that will help you with all of this in KBase, what I just said. And you may have a similar sort of analysis needs. Your data may have, if your data has similar, you can imagine your data, it fits right in. But before you can do any of that, you have to prepare your sample metadata file or import the sample. So this is what this webinar is to help you guide through that process, give you more details. So by the end of it, you will have some idea about what samples is in KBase, how things are organized, and how things work. But in future webinars, you can see how to import amplicon matrix, chemical abundance, and a whole set of cool apps to do statistical analysis, PCA, and get insight into the data. But the cool thing is all the data and the analysis are linked to samples. So that gives you for now, it gives you a great way to look at the data and also include that in your analysis, the sample metadata, but also see how your data is organized. If you are just one person uploading the data, it may not matter much. You already know, this is the data that you have uploaded. But if you are working as part of the team, I'll show you how you can see all the linked data that are corresponding to the samples. And the ontology is linked to it. So it's the first thing. So let's look at the apps. I'm not going to introduce Narrative or KBase. I'm assuming all of you have some familiarity with KBase and this user interface. And so there are apps. If you search import samples, there are import samples. You can import samples from IGSN and import samples from NCBI. Basically, the idea is that you have a sample file. So you create a sample file. There are other ways to do it, but you create a sample metadata file with all the information. Then you pick up a template. You have KBase, SESAR, Enigma 3 templates that we are supporting currently. You give it a sample set name. So sample set is a way to organize samples in KBase. And this is what appears here. You don't have access to individual samples in this space. You have access it as a sample set. And then some sample set description. We have ways to validate the data, and it will give you error. I'll show you how to figure out those errors. And if you know that your fields are not supported, you can choose to ignore warnings because you know they're not supported, they will fail. So in certain cases, you can choose to ignore warnings. So let's look at first case, which is pretty easy, is if someone has already done the hard work, or you have done the hard work, and you have data in external repositories and you want to import, you don't want to, do the hard work second time, then you can just directly import from that system. So the first thing we have prioritized is the SESAR system, which is at geosamples.org. It's the Earth Sample Registration System. And in this case, what I did was I don't have my data at this site, but I searched data that Pamela had on this website, and I was able to find data sets from her. Normally, you would do it if you have your own samples on this website, or unless you really know what these data sets are. And I can get a list of IDs, as well as I can download the entire thing as a file. So if you have data at this SESAR system, you can download it as a file, or you can get those IDs so that you can go to the next step. So let's say if you have the IDs and you're working with the IDs, there's an app import samples from IGSN, and you can just copy these IDs from there, basically paste it in your as a comma separated, give it a name, give it a sample set description, and hit run, and sort of it creates warnings. Like in this case, this particular field is not supported by KBase, so it will give you a warning. Upload succeeded, it worked, but it gave you warning. You could have decided to ignore warnings in this particular case. This is the case where you would want to ignore warning because KBase is not supporting that particular field. So before I go on, let me tell you a little bit about how the data model in KBase. So KBase, when you're bringing data to KBase, it's not brought in as a flat. It's brought in as a flat file, but it's sort of integrated with everything else in the system. And there's a structure to it so that things can be linked within the system. So as you are bringing data to the system, you are actually creating a database within the system itself. So it's a really complicated problem to support such a thing, where you have data from different repositories, different users, different signs come into the system and try to build a data model that can take care of all of those scenarios, given that a bench biologist might upload it, a bioinformatician might upload it, different kind of samples could be uploaded. So it's a pretty challenging problem. How do you build such a system? So one of the things is the system needs to understand, the sample system, what other systems are giving it, what people are uploading it. So it needs this information about mapping between the vocabulary that exists in the external system and mapping that exists within KBase, and it should work seamlessly. And so a lot of effort has gone into supporting particular formats right now, and we hope to increase more and more of such resources into the system. But like in this case, you can see this is the format that is downloaded from SESAR. These are the standard fields that are KBase expects from a SESAR format. And so once you bring this file, you can upload this file into KBase. You can select SESAR format and basically give it a name, again, a sample set name. And basically it will create a sample set again in this. And in this case, upload succeeded, there were no warnings. And it creates this sample set object and links to samples within the system. So that was how you would upload data from SESAR. So let's say you have data at NCBI BioSamples. We haven't yet fully supported it. This is still a work in progress. The fields that are in NCBI are not completely understood by KBase. We have to create that mapping. We have to create the mapping for all the packages. And that's a work in the progress, and it is one of our high priority list. But what I was showing here was you go to bioprojects, you can search for the data set of interest, projects of interest. And in this case, I'm searching a particular things from marine sediment genome. I find there are 155 biosamples in this. I can go to click on that link. I can go to this page where it has the details of each sample. I can then use the summary thing and get all the IDs corresponding to that biosample. I can then basically bring that into KBase. I'll use the app import samples from NCBI, quick cut and paste for all the samples. And KBase will sort of bring all of this, pull this data from NCBI. So you don't have to download anything there. But if you see there, in this case, upload succeeded, but with 21 errors or warnings. There's no error, otherwise it would have failed, but there's a warning. The yellow ones are warning. The red ones would be errors if there were any. Now, these are the fields like publication underscore date or last underscore date. These are the fields that KBase doesn't yet, there's no mapping to that in KBase. There is an equivalent in KBase, but we haven't sort of mapped it yet. So it's still a work in progress. But right now you can bring this into KBase if you don't have any other way to bring that data. And there are ways in which you can modify this data to meet KBase standards. I'll show you in a later slide. So this was the case where someone had already done the hard work and you were just importing data from there. But let's say you are a project that had just started and you want to bring data into KBase, or you're working at a part of multiple team project and you don't have yet data in some repository. How do you bring that? How do you format your data? Where do you begin? What are the control vocabularies that you should be aware of? How do you find things in KBase? And so I'll talk a little bit about that. The first place you start, as Ben said, was the sample page, which is at docs.kbase.us. It has great detailed links to all the information, how to upload different kinds of data sets, what are the formats that we support, the templates you can download from here, the vocabulary documentation page for SESAR. And again, information about NCBI BioSamples, Enigma format that we support, and the corresponding template. The permissions for samples, how you update sample attributes. I will be talking about these right now, but I'm just pointing that these are the information available on the website as well. And you can get that information from there. But this is where I'm introducing control vocabulary. There was a link here, which I'll go back to. So there's a link, most up-to-date version on this list. It's on the using ontology and validated fields. So it brings you to this page. And this is a really, really important page because it gives a list of all the fields that are supported in KBase. So when you're preparing your file, you want your data, the column headers to be from one of these fields. And it also tells you how things are being validated. And so if you know if there's an error, you know how to fix that error. So let me give you a close-up into that page. So that page had, the first column was key. So like name would be a key, latitude, longitude, material, biome. These are the keys. So these are the fields that KBase understands. So if these are the column headers in your file, then KBase would understand it. There's a description of it. And then how it's validated. Like name field is validated, it has to have a maximum character of 240. If it's longer, it will give you error. Number, latitude should be a number within some range. Same thing for longitude, number and some range. Material, and this is where, this is the most difficult part of building sort of the samples, is to find the ontology. Anyone who has worked with ontology knows that sometimes fields are available, sometimes not available. It's sometimes difficult to find that information as to what is the right ontology to use here. But if you're using material as your column header, it expects you to have an ontology field in there. In future, we may have better ways to validate and sort of use this, but right now you have to have this. And I'll show you how to find the child term for this, this particular ontology term, which is needed. Same thing for biome. Biome is also validated. Now, if I'll show you again, if you're working with your data, if it's raw data, you haven't put in any ontology information, it's good enough for you, you know that. But for that data to be really fair and for other people to understand that data, and for it to be linked to other things in the system, it's really, really important to have this sort of ontology information linked to it. So that was some description about where to find documentation, where to go to the control vocabulary page, and what is supported in KBase. Now the SESAR template I earlier showed you, again, sample formats that we support. So SESAR has a template that you can download that looks like this. It has various fields. And there is a vocabularies documentation page. So that SESAR's page, you can go there and make your data according to the specifications of SESAR and upload into that system and bring that into KBase using the methods I showed earlier. And in that case, you will choose your template as SESAR. Before I go into the KBase format thing, it's less restrictive sort of a format right now. But let me introduce the data that I'll be using that for the demo. So Sanj Angleth, who I mentioned earlier, is somewhere on these boats and collected samples from the ocean. And so he shared this data, his raw format that he had the data from. And it was an NSF-funded project, basically. So I'll use that to show what are the steps required to convert data into a format that can be used for KBase import. So imagine this is the data that you have, or SON had. So it had sample ID, lat, which is not standard, long, which is not standard, collection, not standard, cell count, material, and biome. Now, if you are the one who has not generated this data, it's very difficult to interpret what's going on in this, what is the material there. By latitude and longitude, you can make sense. Yeah, this is from an ocean. But other than that, there's not much for you to link to or understand or get insight into this data. But with and if it's lat, the system cannot understand that you're talking about latitude. Given the context, as human beings, you might understand. But the computers can't understand. So it needs exact terms. So in order to convert it, name is the first field. Instead of sample ID, it needs name. Instead of lat, you give proper latitude, longitude. And you again go back to that sample metadata field space that I showed earlier. And these are the fields that are available in there. So you have latitude, longitude, and collection method is something that is understood. Cell count is not understood, but there is a field, enigma : DAPI cell count, that you can use. And then material and biome was not present in this, but this is a seawater. So the ontology corresponding to that was obtained. And same for the biome, it's from ocean biome. So the corresponding ontology is obtained. And this is the simplest data that can be uploaded to KBase, that is well validated. And you can have many, many more terms. For that, you will have to go into that metadata validator page, look at each of the fields, which ones match your data. There might be fields which you won't find there. And we are working on adding more and more templates and fields in future. But let's say you upload this data, you will find errors or warnings. Let's understand the warnings again. So lat here is, it showed in yellow in the report that you saw earlier. And that means that lat is a user-defined column. The system doesn't understand what it is and will not be validated. So you can put a number here, text here, whatever you want to put, system will not try to validate it. And so it won't be able to display it on a map. But if you use properly, like latitude and longitude, then the system can understand and then display it on a map. Similar thing for material. If you don't put an ontology and if you put some random text string, it will try to figure out if it's an ontology or not. If it can't resolve that ontology term, it will give you an error. So now, as I said earlier, finding ontology is the most difficult thing in sort of making this whole data fair. Anyone who has worked with ontology would know it. So just to make things easier, there are these two pages that exist for finding all the child terms that KBase will consider. So if you have a biome field, you can find your ontology term on this page. If it's in this is represented in a hierarchical way. So I was looking at seawater, which has this hierarchy. If your term is not available, you can pick term from something from a higher level as well, but it's better to be accurate. And so for material, you can then, in this case, I picked up this particular ontology. Same thing from biome page, you can find the right biome for data. It helps make things a little bit easier. And hopefully we'll have better ways to find this information within the system itself in future. So you have created samples, you have done all the hard work. And let's look at what are the things like right now, without attaching any amplicon data, chemical abundance, nothing. What are the things that you can do with this data right now? So there's a landing page for the sample set. So this is the object that is created in KBase. And as I said, since it's a validated field, latitude and longitude are there, you can see the map. You can see how the data is grouped, individual positions. If they are overlapped, they will be shown together. You can see a spreadsheet view. This is what you have uploaded. But if you're looking at someone else's data, this view is available. You can also go to individual sample pages and look at more details on what ontology it's connected to and other relevant information from there. You can look at the graph of the ontology, related ontology, and that this particular ontology term, how its hierarchy is there. If there are linked genomes from this environment, in future we'll populate this field. So you can quickly see all the genome that you have are from this particular environment or obtained from this material. It will be linked in this. Any other samples that are linked to this ontology, you can find this. So you can quickly see how it can help you sort of go from your data that you started with a particular ontology that was linked to your sample and find all other linked samples within KBase, either which are public or which you have access to. And that gives you a great way to look at other people's data and look at your data in the context of their data and their analysis. So it makes it really, really powerful. This tab is not yet populated, but we are working on making this tab populated so you can go from ontology to samples. So what I did was I also uploaded, just to show you this, for these samples, I uploaded amplicon matrix as well. And now for this sample page, it shows up in my link data. Now, again, if you are working with yourself, you already know what this is. But if you're working as a part of a team, you can then imagine your sample being connected to your amplicon data, chemical abundance, expression. And as more people are putting in data, and you can see how the measurements corresponding to your samples are increasing and what are the right tools that can be used for the analysis. So it's really, really good for sort of a big sort of collaborative projects. It can really be helpful in those situations. And we are also working on sort of, before I move on, is sort of ways to figure out, easier ways to figure out for you to ask questions like, give me all samples that have chemical abundance as well as amplicon matrix so that I can do the correlation. And if other people are uploading the data, you can easily figure out. So we are working on such apps that will help you find those data sets pretty easily. One thing that is, in this particular case, this page access shows that it's me who has uploaded the data and the system admin who has access to this data. But there are no other people. You don't want random people to sort of, as I said earlier, to edit information in your samples or link random data to your samples. So we have ways to update access control. Before I go into updating access control, let's imagine that you made an error in your upload somehow, and now you have to re-upload the data. So how do you go about doing that so that the system doesn't create new samples, but sort of links it to the earlier sample that are there and creates a new version of it, so that things are linked and not And so in that way, you can update with new information with the sample. So you will use, I don't think I have a screenshot here, but you will use the import samples app, and there's a field sample set. That was the first field. So you provide an existing sample set as an input there, and it will look at the samples and figure out how to update it properly. So now you see in this particular case, there were all the new versions, version 2 of it. And in this particular case, I had added information about the collector who had collected the sample that was missing in my first iteration of upload. I can see all the versions of this data and basically figure out how this sample information has evolved over time. So the last one that I'll talk about is updating sample set access controls. Basically, in this case, you select a sample set. You have to specify the usernames of people that you want or are on your team, and you're confident they won't mess up with your data. You put their name in. You give them specific privilege if you want them to be admin, writer, or reader. And so then you have control as a project, as a team over the samples and what can be linked to those samples and who can upload relevant data. So that was all I wanted to talk about samples. Hopefully, it gave you a sense of where to start in KBase, what are the resources that we support, where to begin uploading data, what are the apps that exist, and you have a little bit more clarity on how to upload samples in KBase. And so this was the purpose of making these modular talks, so that the first one was overview, the next one goes into a little bit more detail. But hopefully if you have samples, you should start uploading, go to the documentation page so that you can be prepared for the next set of talks, or if you have questions, connect to KBase. We have Jira account that Ben can guide you to. But in future, we are the related talks, related samples are samples and amplicon analysis, the cool analysis, the set of apps that Pamela and her team has worked on this. You'll hear from her. And then samples and chemical abundance analysis. And Janika will talk on November 10th. And using samples to link multiple forms of measurement and correlation analysis. This is where you will get a lot of insight from your data. And so these are the seminars we have in progress. And this is the team that has closely worked with the samples team. I mean, in general, a lot of people in KBase have contributed directly or indirectly to this effort. But these are the more direct people who almost meet on a regular basis and have been thinking about the design and implementation of KBase. With that, I think, thank you, everyone. I don't know if there are questions, but.
Thanks, Ranjan. Yeah, I don't see any open questions in the Q&A doc, but maybe there are some of you with verbal questions that you'd like to ask. So what I'm going to do now is allow participants to unmute themselves. And if you want to ask a question, you can, or if you want to raise your hand to ask a question, I can call on you. But yeah, I don't know if there's any verbal questions that folks would like to ask while we've got the KBase team online.
Yeah, we have many of the KBase team members, samples team members on the call.
There was a question in the Q&A about creating custom templates, or if there's a way to request new templates. I saw there was an answer in there, but maybe you've got some thoughts on that, Ranjan.
Yeah, so custom templates, there needs to be some work done on it to make work in KBase. We don't want a proliferation of terms. And so the ideal way would be to work with KBase when the time is right, to sort of find the mappings that already exist in KBase to the mappings that to your mappings. And we have some thoughts on how to make that possible. It's just that it's part of the more later version of our sample effort, and not sample 1.0. So maybe sample 2.0 would have those features. But if there are fields that are missing that you think should be in KBase, please feel free to reach out to us. If you have a sample at NCBI and you want to bring in, let us know the kind of samples that you're interested in so that we can prioritize those packages. So yeah, Jira is there. Create tickets on Jira, let us know. And it'd be great. It's just the start. It's our first, introduction to samples. So it'd be great to get feedback from the community and see where things are and what we need to improve.
Hey, this is Shane Kim. I'm one of the managers for KBase and helping sort of lead some of the samples work. So just to follow up on Ranjan's point there, I think what we're looking for is we prioritize templates that we think kind of represent a broader community so that, there's a bigger win there. And then long-term, we're trying to figure out a model where people could easily kind of bring in their own. But like Ranjan said, that'll probably be in a later iteration. But we're interested in finding out, we've identified things like SESAR and BioSamples. If there's other, kind of large sample repositories that people believe are important and we should be kind of watching and tracking and looking at, we'd certainly like to hear feedback. You can put it in the Google Doc, definitely respond like Ben was saying to the survey. There's some questions in there around this, or file a Jira ticket. And we're happy to have some, follow-up conversations on that if people want to discuss it.
Thanks.
Great. Yeah, I just dropped the link again in the chat to that sample survey if the folks on the call are interested in filling that out. Yeah, I see there's some more questions in the chat. Or in the document, someone asks, sounds like there's ongoing work to map from INSDC biosample. Does that include GSC MIXS terms?
Yeah, so the NCBI biosamples, we will support MIXS templates, but if there are specific MIXS templates that you're interested in, reach out to us, let us know, and we can prioritize those.
Yeah, there's a lot of different packages, and there's also a lot of, their vocabulary is quite large, and we want to eventually pull all of those in, but it'll take us a little time. So if people have particular ones that they think are important to get in sooner, we'd like that feedback for sure.
That's something that we are working actively on, bringing NCBI samples data. It's a great time to sort of connect.
Got another.
And be our beta testers if you're interested. That would be great. We are looking for beta testers.
Yeah, there's a link in the Q&A that I'll just drop in the chat as well for beta testers. And there's another question here. I might have missed it, but is Enigma the best template to use right now if you have lots of data from subsurface wells?
It is more supported. It has the fields that are understood, but not really. I mean, I'll let Shane answer that.
Yeah, I think we did the Enigma template because it was a project we already were working with quite closely. I think long-term, it's something that might be an example of a custom template that we would allow people to generate and use that way. It does have a lot of predefined fields that probably are relevant to well type surveys, and so definitely look it over, look at the terms that are defined there. But like any of these, if you see gaps in what you would like to have fields for, let us know. And maybe one general other comment is we're still working through kind of like what attributes are inherently part of the sample and which attributes are some other form of measurement that might be linked to the sample. And so certainly the amplicon data and the chemical abundance data are two examples like measurement data that are linked to the samples. I think there's others where it's a little bit fuzzier. And so, if people come to us with terms, part of the conversation is trying to figure out, like, is this something we need another measurement type or another object type to capture these types of these fields, or is it something that makes sense to just put into the sample attributes? And it could be that that's something that changes over time as well. So it's definitely an evolving kind of picture, I would say.
Yeah, it's definitely a work in progress, but it has a lot of the fields that Enigma people would be interested in. So, as Shane said.
Okay, thanks everybody. And we will stay in touch about the next in the training series. But thanks again for logging in today, and have a great rest of your Wednesday.
Okay, thank you. Bye-bye.
Thanks for watching this webinar. For more webinars and tutorials, subscribe to our YouTube channel. You can see announcements for upcoming webinars and recordings for previous webinars on our website at kbase.us/learn. Let us know in the comments what content you'd like to see in future webinars. Be sure to follow us on Twitter at DOE-KBase. And if you have questions or encounter an error when using KBase, please contact the help desk.